Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Today, I'm joined by Dr. Michelle Halls, Associate Professor at Monash University's Institute of Pharmaceutical Sciences. Michelle's story isn't just one of scientific depth, it's a look at how curiosity, timing, and collaboration can shape a career in receptor biology. From early memories in her dad's formulation lab to her current work exploring how GPCRs organize and signal with stunning sensitivity, Michelle brings a powerful perspective on what it takes to do meaningful science. In this episode, you'll hear about how cells use signaling organization to fine-tune GPCR responses, what makes the Monash model a standout in collaborative pharmacology, and how pre-assembled receptor complexes could change how we study and target disease. If you're serious about sharpening your pharmacology edge, Terry's Corner is where you go. Dr. Terry Kanakin brings decades of expertise to a structured, on-demand learning series built for scientists who want to drive real drug discovery impact. Details in the show note. We are excited to welcome Revity's pH-Sense reagent into the Dr. GPCR ecosystem, a breakthrough toolkit designed to make GPCR internalization studies simpler, faster, and more scalable. With 4 flexible formats, no-wash protocols, and TRF detection, pH-Sense gives researchers the ability to monitor internalization in real time, even at endogenous expression levels, without imaging. Looking for non-radioactive ways to track ligand-receptor interactions in real time? Celtera's research is advancing GPCR science with fluorescent ligands designed for live cell imaging and high-resolution pharmacology. We're excited to have them as part of our community. Learn more by clicking on the link in the show notes. And now, let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR. And today I'm very excited. This podcast has been a long time in the making. I think I canceled it at least once. So this is supposed to be in 2024, but we're now in 2025. And I'm delighted to have with us Dr. Michelle Halls. Michelle, welcome.
Thank you for having me.
I'm very excited to have you on. And for those of you who don't know you, you're in Australia and it's 8:40 something in the morning, whereas it's 4 something in the afternoon here in the Boston area.
Yeah, it's actually quite a good time for a meeting with someone in the US. I've already had a meeting this morning, so we're on a roll.
Wonderful. So let's start at the beginning. Could you please introduce yourself, tell us who you are, and what is it that you do?
Sure. So I'm an associate professor at the Monash Institute of Pharmaceutical Sciences at Monash University in Melbourne, in Australia. I'm a lab head and I'm the deputy theme leader for the drug discovery biology theme there. So my background is molecular pharmacology. What I'm really interested in is signaling of GPCRs and how that's organized in cells. So we're fascinated to try and understand how that's organized, how cells use that organization to get very specific responses, and then how that can change in the context of disease.
And how did you get into studying GPCRs? I mean, I have to say, I typically don't do it. I try not to get too deep into the career trajectory of my guests because I want to be able to ask those questions. But right before we joined, I sent you a LinkedIn connection. And I saw that you've been at Monash and in molecular pharmacology for quite some time. So how did you get into the field?
Yeah, so I always liked science and maths at school. And my father was actually a chemist, a formulation chemist. So he did a lot of optimization manufacturing for things like sunscreens, air fresheners, things like that. So I do remember when I was very little, I think I was sick from school. He took me to work and I was allowed to use the balance in the lab. You wouldn't be able to do that anymore. You had to be careful of the air conditioner flow and to make sure you were accurate with the weighing everything. So I think that helped. For my undergraduate degree, I did biomedical science, which was a really good degree, I think, because you got a really sort of basic fundamental understanding of a really wide range of the sciences. And one of the electives I did during undergraduate was pharmacology, and it went from there really. So we had lectures from Roger Summers on GPCRs and cyclic AMP signaling, and I think I just fell in love with that idea of G protein coupled receptors and specifically cyclic AMP signaling. I just that pathway had me sold. So we had, in our third year, we had an opportunity to do a research placement as a subject. And I never thought I would like research at all. I thought it would be boring and not something that I'd like to do, but I did that project and I just really loved the discovery side of it. So doing something new, designing an experiment, graphing the results, seeing what happened and deciding what to do next, that just really captured my attention. So after that, I did an honours year. So I'm not sure if you're familiar with the honours year in Australia. So it's a small research project that you can do at the end of your undergraduate studies. It's basically 5 or 6 months in full time in the lab. And at the end of the year, you write up a mini thesis and you do a final presentation. Some students do a thesis defense as well. So it really gives you a small taste of what it would be like to do a PhD or to have a career in research. And again, I just really loved that whole process. So I decided to do a PhD with Roger Summers. So that was on GPCRs, obviously, relaxin receptors, and understanding their cyclic AMP signaling pathways. And then after my PhD, I did a postdoc at the University of Cambridge in the UK. So I went to the lab of Dermot Cooper. So he's an expert in cyclic AMP signaling. So this was a step away from GPCRs, more into the fundamentals of cyclic AMP signaling. I learned how to use threat-based biosensors targeted to different areas of the cell, and things like isolating and identifying protein complexes that regulate cyclic AMP signaling as well. So I spent about 2.5, 3 years there, which was a really fantastic opportunity and a great time. Met some amazing people that I still am in touch with and collaborate with now, and we're spread all over the world. So that was a really unique and fantastic opportunity. And then, so I was there on an Australian fellowship, which they don't have anymore, unfortunately, that provided 2 years funding for someone who had just finished their PhD to go and do a postdoc internationally. And then you had to come back and spend 2 years at an Australian laboratory. And the idea was knowledge transfer. So you would go away and learn something new, and then you would bring that perspective or technique back to Australia. So the 2 years back in Australia, I moved to the Monash Institute of Pharmaceutical Sciences there. And Roger Summers, Arthur Christopoulos, and Patrick Sexton had set up the drug discovery biology theme while I was overseas, and it's really just gone on from there.
Wonderful. You were telling the story as to how you first heard about GPCRs and signaling, and the fact that you spent time in the lab, and that very much resonates with how it happened to me. With a couple of differences in names, but that was pretty much it. I told this to anyone who's listened to the podcast multiple times. You're going to say, "Oh, here we go again." But it was very similar. I was sitting in undergrad in a class taught by Michel Bouvier, where it wasn't even a PowerPoint at the time. They had, I don't know how to call it in English, the overhead
yes. Transparencies or whatever that they would draw on. Yeah.
Exactly. And he was drawing on it and things like that. And I thought, oh, wow, this is really actually cool that, you have a finite number of GPCRs, an even smaller number of G proteins, and yet you have this diversity of signaling. So I applied to spend the summer working in his lab. And he ended up referring me to somebody else who was just starting their lab on chemokine receptors. And I spent the whole summer trying to induce single point mutations using the single-stranded DNA Kunkal method of mutagenesis, because at the time there were no kits to do this. So it took me 3 months to generate 6 mutants. And then I spent my master's thesis characterizing these mutants, which I think was a really fun time to be in the lab.
That's amazing. That takes me back, actually, talking about how you were doing the mutagenesis. I remember in Roger's lab during my PhD, we'd be doing cyclic AMP assays, and it was the alpha screen cyclic AMP assay in the 384 well plate, but we didn't have multi-channel pipettes or anything like that. So we'd be using a single pipette, going from one well in the 96 well plate to one well in the 384 well plate, trying to remember where you're at to transferring it. We'd spend hours sitting there with trays of ice, transferring one by one the samples to a 384 well plate. So times change.
Yeah, they absolutely do. I do remember trying out the alpha screen, which, by the way, never worked in my hands. But back in the day, I had initially learned how to measure cyclic AMP production in Michel's lab using radioactivity, where you had the columns. So we had, I can't remember, someone in Michel's lab knew someone who had this very nice way to make a tray to put the columns in. So I packed my own columns, and you had one set over the other, and you had to go down to the counter. And then they came up with the EPAC biosensor, which was a blessing because he was
I know.
and he didn't have to deal with this. I think by that time we did have multi-channel pipettes, but they were not electronic; they were handheld, 8-channel.
Yeah. We eventually had the handheld ones, but that's actually how they still measured cyclic AMP in dermats. Like when I moved there, was with the radioactive assay. So they were doing it was interesting contrast, I suppose. They had some of their experiments, which were their EPAC biosensors, microscopy, but most of their sort of standard cyclic AMP assays are still the radioactive ones.
I love it. Love it. Some people will be listening to this podcast episode and be wondering, what are you talking about? What is that thing? I think it was fun times. It was very minimalistic. Lots of work went into it, but I think it also tested our commitment to try and find those answers.
I think it's interesting. I think that the challenges change, right, as techniques move on. So, we spent a very long time probably doing something that could be done very quickly now, but I think the challenge now is maybe asking the right question and not just doing stuff because you can, like really thinking through what is going to give you the answer, or if you're going to do a lot to try and see what's going on, how do you analyze that most effectively to find something at the end? It's interesting how it changes.
Yeah, I like that. I think you're absolutely right. So at the time when you cloned the GPCR, you ended up having a Science or a Nature paper.
Yeah.
Think about what it takes to get there. And to your point, now we can acquire lots of data very quickly. And to your point, it's very important to ask the right questions, but also to have the right tools to analyze that data, because the human brain cannot assimilate all that information anymore.
No. Well, that's where we hope, right, that AI is going to make a really big difference in terms of, if it can be applied. And again, you would have to be very careful about the questions you ask, but then hopefully start to pick up patterns or similarities or differences that we just wouldn't be able to do if we were trying to do it ourselves.
Yeah. Well, one thing with AI is if junk goes in, junk comes out, and I think that's where it gets interesting. And I think working with computational biologists, computation chemists, or machine learning engineers, the challenge there is to develop that same language and be able to understand each other. I see it on a daily basis where we use the same words, but they don't mean exactly the same thing. So we spend a lot of time.
It's fascinating, isn't it?
Yeah.
Even among sort of what you would consider, I suppose, more biological sciences, you get that. So speaking to collaborators who have a cancer biology focus, for instance, or an immunology focus, you think you're speaking the same language, but you're not really at all.
No,
it's quite a skill.
I think so too, and it's very interesting when I look at people's profiles, and two people can say, "Oh, I have a GPCR pharmacology background," but you actually have to dig deep, and to understand what is it exactly.
Exactly. So I describe myself as a molecular pharmacologist, but in the sense of cell signaling side of that and not analytical kind of side of that, I've got many colleagues that are much better at that side and know a lot more than what I do. So you're right, with get into the details.
Yeah, exactly. What I wanted to ask is, once you obviously very quickly fell in love with GPCRs, fell in love with research, what was the next step after undergrad, after your honors? I'm assuming you really like the lab, you really like the topic and cyclic AMP as well, and you trained in the UK. But did you at any point think about, oh, maybe I should go into industry, or I will stay in academia, or what did that look like for you?
I think, to be honest, at that stage, I didn't think about it too deeply.
No one does.
No, I enjoyed what I was doing. The postdoc was a really fantastic time, I think, because I went there with a fellowship, I had a little bit more freedom perhaps to explore a little my own project in the context of what the broader lab was doing there, which was really fantastic. It's a period of time where you get to focus on the research without necessarily having to worry about where the next funding comes from, or how you're going to support your team members, or what grants you're going to apply for next. So that was really fantastic. And I think I just followed opportunities as they came along. So I didn't ever set out, excuse me, when I was a PhD student thinking what I want is to run my own lab. I want to be a group leader. I want to be doing this. I know there were some students who went through at the same time as me, and that was what they really wanted. That was never something that I specifically thought that I wanted, but following the opportunities as they came along kind of eventually led in that direction. I'm not sure if that's very helpful answer.
No, I mean, a lot of people, to be honest, anyone I talk to gives me the same answer as you just did. I did the same thing. And, you read this self-help or these books where they tell you need to plan ahead and you need to make a plan and figure it out. And what I've been realizing is that it's very difficult.
Yeah, I think that's good to do. I think maybe that's probably something a lot of us don't take enough time to do, is to take that time out and kind of forward plan and think about what we really want to be doing. But I think you also have to be flexible. I think particularly, probably all professions are the same, but my experience is obviously in science is that, you can apply for 10 grants and if you're lucky, you might get 2 or 3, or you submit all these papers that you think are going to be amazing and, 50% of them get knocked back. And so it's doing what you really love, keeping on doing that, taking opportunities as they arise. And it might not be in the timing that you wanted or in the order that you wanted. But hopefully if you can do that for long enough, and if you're supported to do that for long enough, because I don't think you can do this by yourself, then you can, the opportunities arise to get to where you would like to be.
No, I agree. I agree. And I think you can't do it by yourself. I think as long as there's a direction and you're aligned with that direction. So in your case, in my case, it's GPCRs and it's molecular pharmacology. Whether it's in academia or industry, as long as you can make an impact and you can find answers or even think about what are the questions that you're excited about, the rest is details.
Yeah.
At that point.
Yeah, exactly.
So how is it now that you're leading a group and you're working with so many people? And I know the Australian or the Monash ecosystem is a very unique one in the world. I've had Patrick, I've had Arthur on the podcast, so I have a sense of what that looks like. But I'm curious about, from your point of view, from the time you were, a PhD student to now, how did that ecosystem evolve and how do you fit into it today?
Yeah. So as I said, I did my PhD with Roger Summers. So we were out at the pharmacology department at the main Monash campus in Clayton at that time. During my PhD, Arthur and Patrick and their teams moved to the same department, and Roger and Arthur and Patrick started working together. And so then I left to do postdoc, and in the meantime, they moved to the Monash Institute of Pharmaceutical Sciences, which is in the Parkville campus, closer to the city, and really started to build the drug discovery biology theme as we know it now at that point. And so I think this is kind of what I was coming to before in terms of support. And I think also being lucky, right place, right time. So there was a whole group of us who were doing PhDs around the same time. So Lauren May, Karen Gregory, lots of other people at Monash, and people who came to do postdocs like Celine Vallant, Denise Wooten, Greg Stewart. And we were very fortunate that Arthur and Patrick and Roger were really supportive of us trying to do what they could to help us become established as research leaders. So we had, in the beginning, a shared funding model. So anybody who got funding in, it would all go into a pool and we'd share it around. And this kind of bridges the gaps that you get in a lot of grants where the salary for a postdoc is not actually enough for a proper salary for a postdoc. And so I think that was a really fortunate time, and it really helped to build that system we have there. So we have a lot of shared lab spaces, shared equipment. We contribute together for a lot of things. So I think it's really seeded that model almost of collaboration and working together, pulling towards a common goal rather than lots of individual groups doing their own thing.
I think it's wonderful. And I think, do you think it's because of the fact that, it's in Australia, you're far out from everybody else in the world? But I love the system because it allows you, as you said, to pool your resources, to share the instruments. You need 2 pieces of instrument, and then everybody else can use it. You share the responsibility of maintaining those, but at the same time, everyone has access to them.
Yeah, I'm not sure whether it's because of our location or maybe partly that, and also perhaps they had an opportunity to design the labs when they moved over, and there were a few people in that department there already, but really when those groups moved over, they were the bulk of the people. So I think that really gave them an opportunity to set it up in a way that they thought would work really well. And it was effectively one big lab at that point, but then has grown into lots of individual labs, but still has that feeling about it, I suppose. Yeah.
Wow. So it's this large open space, and now things are segregated by bench. I'm assuming this group has this many, and
so and it's something that we have that sort of space is reallocated on a yearly basis depending on the number of people in each group. So that obviously always changes with students coming and going and postdocs coming and going. So it is fairly fluid, which I think is good as well, because you don't get that demarcation of this is my area, you can't go there, all that sort of thing. It is quite a dynamic organization, I guess.
I think it's really nice as well because, if everybody's in the same space, you get to have conversations, you get to hear somebody talking about something and you say, hey, well, I had that issue, I can help you with that.
Yeah. I think the other thing that helps with that is the PhD program we have at Drug Discovery Biology as well. So it's in a way based more on the US model than what we would typically have in an Australian PhD, in that students are accepted into the PhD program. They do coursework in their first year and they rotate around 3 different groups to do 6-week short projects. And because all of the students start at the same time and rotate around these groups together, they build a cohort. So they already know a lot of other PhDs in the same time as them, I suppose. And they also build connections in the different labs. So by default, they already know someone who does this really well in a different group, so they can go talk to them. And I think that really does encourage that continued connectivity, which might otherwise have just been kind of at the group leader level at this point, right? But that helps that the students have that coming through, and then the postdocs end up doing that as well.
I love this model. So you have to make sure that the students are happy. The students have to make sure that they've done a good job, because word also spreads around very easily, positive and negative.
That's true.
But I think it's great because it's transparent, and then it leaves no surprises.
I think it's good for the student and for the supervisor, right? Because the student can decide also, well, I might like this topic, but this lab environment in terms of the way they run things or the other people in the group suits me better. And it does also then start to encourage collaborations or co-supervisions on projects that, well, I want to do my PhD here, but I would like to take an element from there. Let's work together and move that project forward. So I think it works really well.
And to your point, it's not something that in a traditional setting you could be doing, because you'd be maybe in the same corridor, but it's 2 different labs. And you would have to, I don't know, juggle with the 2 professors and say, well, I want to do this and you're doing that. Can we collaborate? And again, I feel like there is this extra energy barrier that you need to overcome. Whereas in a system like this one, it allows you to very quickly say, well, I'd like to do this and that. Can we collaborate?
Yeah. That's great.
Wonderful. In your group, how many people are there, and how do you typically, what's your management style from a scientific perspective?
That's a good question. Now you're going to have to make me count on my fingers. How many people do we have? Must be about 5 or 6 at the moment. So I have a postdoc, Alistair Keane, who's wonderful. Vernon Liu is a research assistant who's been with me, but I don't even want to say how long because it reveals too much. And then we have at the moment 5 PhD students, another one just about to start, and then typically we'll have maybe 1 or 2 honours students during the year, or because we've got this rotation model, we'll be hosting rotation students throughout the year, as well as undergraduate students and master's students on project placements as well. In terms of my management style, I'm not sure. So I meet with my team regularly. So we have one-on-one meetings once a week or twice a week once they get further into their PhD. We have lab meetings. I really like them to take ownership of their projects. I love it when they come to me with new ideas or ways of doing something, things that I haven't thought of, and try and I see my job as trying to facilitate their research and their success as much as possible. So, what does this particular person need to feel comfortable, feel confident, to excel in what they're doing? And that might be very different to another person. So, trying as much as I can with the resources and facilities that we have to make sure that each student gets the most out of their PhD, or each postdoc gets the most out of the time in the group, and can we make sure that we're setting them up to where they want to go next, I guess.
No, I think that's wonderful. And to your point, not everybody needs the same type of supervision. Some people need a little bit more hand holding in the beginning. Others, you can let them go and they'll come to you when they have questions. And I think evolving into this supervisory or not managerial, but more leadership role involves recognizing when to push and when to let people just go. And it's a tough balance to do as well.
It's really hard, and I think it's taken me a long time to learn that as well, because when I was a student and a postdoc, I just wanted to be left alone to do whatever I needed to do, and then I would come and show you when I had results or when I had a question, and that suited me perfectly. So I think that was my default setting when I first started supervising people, but I quickly realized that doesn't suit everyone. Some people are much happier if you're checking in more often. Some people need more constant kind of reminding, we need to be doing this now, we need to be doing this now. It's just different styles, I suppose, of doing the work.
No, the reason I was mentioning this is because I think last week I was talking to a colleague, and in my, let's say, if you come to me for help, I will drop everything and help you. And I realized that I needed to work on this. And so for the first time in a very long time, I said to my colleague, I said, can you give me 5 minutes? I'll be right there. And I finished when I needed to and went to talk to them. But it also reminded me as a PhD student, I was actually transferring something from one 96-12 plates to another when my PI came and he was asking me questions, and he loved my reaction, but he was so honest. I looked at him actually halfway and said, not now. Because I was so afraid to mix up the thing. He was so important, and he loved it. And I said, well, I think I should be more authentic and say not now. And apparently that story went on even after I left the lab. I was one of his first PhD students, actually his first PhD student ever. And even after I left, he used to tell this story to everyone. You mean I said not now? So I think
it's particularly when you're in the lab, and I think particularly senior students, figures in the lab, often the more junior people will go to them to ask for help all the time. And I'm not in the lab a lot anymore, so I think it tends to fall to the other people in the group. So for them to be able to say not now, I think is probably quite important. And it also gives the other person a chance to go away and think about it for a bit themselves.
Exactly.
For chatting to some colleagues at work, I think they've started to encourage students to say, well, come to me if you have a problem, but also come with a possible solution, right? So that you're not just asking the question because it's easy and you're going to get an answer. You've thought about it. I've got this problem. This might be what's going on. I think we should try this, and then start the discussion from there. I think that's a really good idea.
I love it. No, absolutely. I think that's also to encourage. I do that with my kids. What do you think? My question to them is, what do you think we should be doing?
Yes, I do that too.
They're different types of questions. They're not necessarily scientific, but I think that could be used as well. Let's get a little bit back to the science. I asked this from everyone. Sure. What's your favorite GPCR?
I don't think I have a favorite GPCR. So I would say anything that's got interesting signaling. So I would always have a preference towards cyclic AMP signaling, but something signaling that's localized to a small area of the cell, or a receptor that's got a protein complex that's really interesting that controls what it does. I think that's really fascinating. The organization that surrounds how GPCRs work and controls their signaling has always fascinated me, whether that's organization in terms of location in the cell, or organization in terms of interactions with other proteins and scaffolding and recruitment of different effectors. I think the complexity of that I find fascinating.
Yeah. What I love about it is not only it's complex, but it's also finite and time bound. And there's a kinetic to it.
Time in there. Yeah, exactly.
It's just fascinating. So I typically leave these questions towards the end, but since we're talking about, localized signaling and cyclic AMP, what are the top 3 aha moments that you had as a scientist during your career trajectory, that got you to where you are today?
That's really interesting. I think the first one, I suppose we were just talking about there, is that whole idea of signaling being organized. I think during my PhD, reading some of those first papers on GPCR, cyclic AMP signal organization by Paul Insel and his team, just really fascinated me. The idea, well, back then I suppose it was that receptors could be in lipid-rich versus non-lipid-rich domains of the membrane, and G proteins would preferentially go to one or the other, and this was a mechanism for specificity and control. I really liked obviously I like the idea of organization. I really liked that idea. So I think that would have been the first one. And that's really what inspired me to go and spend time in Dermot Cooper's lab, because he was one of the world leaders in terms of calcium regulation of adenyl cyclases. And in terms of calcium stimulating adenyl cyclases, there's only 2 cyclases that are stimulated by calcium, and it depends on calcium bound to comodulin, and it depends on capacitive calcium entry into the cell. So there's a lot of organization there, and ACAPs and scaffolding proteins bringing these 2 signaling pathways together. So I think that would be the first one. The second one, when I was in Dermot's lab, I was still working on relaxin receptors, but using the EPAC biosensors to look at the cyclic AMP signaling in more detail. And I kept seeing cyclic AMP responses to really, really low concentrations of agonist. So typically, sort of the bottom of our concentration response curve for relaxin would be 1 picomolar, and that was it was consistently above the vehicle response every time when we were measuring cyclic AMP at the plasma membrane. And so I just kept going a little bit further and a little bit further and a little bit further and a little bit further, and we could still see cyclic AMP responses down to femtomolar, attomolar concentrations of ligands, so minus 18 molar. And that I just found fascinating, and I was fortunate enough that I was able to follow up on that during my postdoc, and we found that these receptors seem to pre-assemble with a really large protein complex at the plasma membrane. So ACAPs, adenyl cyclases, beta-arrestin, PKA, PDEs, everything is kind of pre-assembled with the receptor, and somehow this is creating this ultra-sensitivity to ligands. And we were able to follow up on that when I went back to Monash, and it seems, as far in our hands, it seems to be a common property of a lot of GPCRs. So the beta-2 adrenal receptor does this, the M3 muscarinic receptor does this. We don't know why or exactly how at the moment, but I find that idea that somehow perhaps the conformation of the receptor that's being induced by these interactions with other proteins, perhaps it's opening it into a superactive conformation. And it seems that only 1 or 2 receptors are active with mathematical modeling. 1 or 2 receptors per cell are enough to activation of those receptors is enough to induce a signal. And I just find that really interesting, the idea that receptors interacting with different proteins can completely change their behavior or their classical pharmacology. And then the third, I think, is really something we've been working on for a while now with the collaborator, is looking at how the signaling of GPCRs can change with the onset of disease. So specifically, we've been looking in triple-negative breast cancer, and we collaborate with Erica Sloan on this. She's also at DDB at Monash. She's got a cancer biology background, and she did some work during her postdoc in the US showing that activation of beta adrenoceptors, turns out it's the beta-2 adrenoceptor on triple-negative breast cancer tumor cells, by stress can accelerate cancer metastasis. And so I became really interested in this, and we started to look at the signaling of the beta-2 adrenoceptor in an isogenically identical cell pair. So we had parental cells and then a highly metastatic variant of the same cell line, and we saw completely different signaling from the beta-2 adrenoceptor in the 2 cell types. So in the less aggressive cells, we just get activation of normal cyclic AMP signaling. In the more aggressive cells, the cyclic AMP seems to also influence calcium influx, and it drives accelerated invasion. And we've got some preliminary data that still hasn't been published suggesting that we get differential organization of the receptors on the cell surface in these 2 different cell types. So I think it's a chicken egg situation, but my hypothesis at the moment is during that sort of acquisition of a more metastatic phenotype, or during EMT or whatever happens in the tumor, we get such a big reorganization in these cells, like the cytoskeleton changes, proteins move around, and somehow this is allowing now the beta-2 adrenoceptor at least to couple to sort of more pathological signaling. And so this change in the cell allows activation of a receptor that was probably doing something very nice in the normal cells to contribute and accelerate disease progression. So I think that's maybe an area that's in general that fascinates me, because in the drug discovery development process, we're typically doing things in a cell line with overexpressed receptors. But what happens when that drug hits a receptor in a pathological condition? Maybe it's not signaling the same way anymore. Maybe it's doing something different. And I think there's opportunities there if we understand a little bit more about what happens to these receptors as the cells and the tissues around them change.
I had goosebumps throughout all three because I think it is really fascinating. It is absolutely fascinating. If we go back to the number one that you had, the fact that you had this lipid raft reorganizations and lipid environment changes receptor signaling and receptor ability to signal. I always thought it was fascinating, but at the time when this concept came out, there was little data and there were not a lot of tools. To be able to study that and say, okay, more cholesterol, less cholesterol, more of this lipid, less of that lipid. And I think we're getting there slowly but surely.
Yeah, I think all of these sort of breakthroughs in understanding and knowledge depend on the tools and the instruments we have to measure them, right? So you can have all of the most wonderful theories in the world, but if you can't directly show that this is happening, it becomes really challenging. And I think we're at a really good point at the moment. We've got some amazing tools available to start to look at this. And I think that's only going to get better, and to allow us to really start to quantify some of these changes and these signals in different compartments. Yeah, it's fascinating.
I think so too. And unfortunately, as you mentioned, we typically do this in a Hexel where you transiently express and then you look at something, but then, you may try to knock out a G protein, but then, you have one GI alpha I1, but then I2 comes in and rescues the day. Or if you want to study, receptor activity modifying proteins like Rafts, for example, good luck finding a cell line that doesn't have that. It's very
hard,
difficult.
isn't it? Because you have to start somewhere, though. I mean, I know there are limitations to your standard cell line with transient transfection, but you need to know what you're looking for before you go into those more complicated systems, right? So that's also really, really important. I wouldn't want to say that's not important, but it's also interesting how expression level and exogenous versus endogenous expression can really change how these receptors behave. So where they are at what point in time, what's regulating them. So we typically, and I know lots of people see this, if you exogenously express a receptor, you might try and measure, say, cyclic AMP with the EPAC sensors and you see a sustained signal. Whereas if you measure endogenous receptor activity, you get something that's much more transient. It's just very interesting. Yeah.
No, yeah, absolutely. The point I was trying to make is that it's important to have that starting point, but it's also important, as you mentioned, with the breast cancer cell, to have that disease model. And then ideally you want to take it one step further and have an animal model.
Yeah, exactly. And that's where that collaborative network becomes really important, right? Because you just can't, I can't do all of that myself, that's for sure.
No, you can't be everywhere, for sure. And I think being in an environment that's conducive to collaborations, I think it's the best place to be.
Yeah.
All right. So we've talked a little bit about your scientific interest, how you got to where you are. Also, your interest in receptor complex organization, very unique signaling. I haven't spent enough time thinking about the relationship between cyclic AMP and calcium, and I think I'm going to start looking into that because it's very interesting. You might think that GS is cyclic AMP, GQ is calcium or IP1, and those two don't speak to each other, but they actually do.
There's so much crosstalk between those two pathways at multiple different levels. Yeah, it's a really fascinating system to think about.
It is really awesome. If you had advice for junior scientists who wanted to contribute to the field, what would that be?
I think we've probably covered a lot of this already, actually. I think it's really important to do something that you're fascinated by. I think a PhD, science is hard, but it's fantastic, and we can have a lot of flexibility in what we do, and we're discovering new things, but it's also really hard. So when you're having those hard days or weeks or months or whatever it may be, if you're ultimately working on something that you find truly fascinating, I think that makes a really big difference. I think that's why most of us are here. We're not here for the fame or the glory or the money or the pay or anything like that. So I think that's really, really important. I think it's really important not to underestimate the element of luck that's involved in this career. I've seen so many wonderful scientists that things just didn't necessarily go their way at the right time, who are not in the career anymore. I think it's not always obvious, and I know we were chatting about this before, Yamina, but the people who are still here have more often than not have had something lucky fall their way at the right time. I've had this myself, you apply for a lot of fellowships or grants, and it really needed to get up at that time, and it happened too, but it could easily have not have, and then it would be a very different story. So I think it's important to acknowledge the role that luck and timing can play. in our
Absolutely.
careers.
No, please.
And I think that, oh, sorry. I think the final thing is it's really important to pay attention to what you're doing, particularly with that attention to detail when you're in the lab. Those observations, saying, oh, that's a little bit odd, and not dismissing it, because that might be your discovery. That might be the next Nobel Prize over there, right? It's the things that are unexpected that you didn't think that you would encounter can often be the most interesting, at least I've found for me. So making sure you're paying attention all the time and follow up on those strange things that happen, because you don't know where it's going to lead you.
No, I agree. I've had, actually, my PhD supervisor was mentioning that his PhD thesis came out right out of the trash because something didn't work, and he said, this is trash, and then went home and thought about it, said, wait a minute, no, I have to reanalyze this and take a look at it again. But to your point, I think being able to set up assays that are reproducible, where the positive control is positive, where the negative control is negative, is one thing. On the other hand, I've worked with chemokine receptor splice variants, and the number of times I repeated that EPAC assay, because every time I would get something funky, and I had to do it 6 times and make sure that everything was well, that allowed me to then say, well, this is how the receptor functions. But I needed to follow up on that. Otherwise, I can't say anything.
I think, I suppose, further to that a little bit is we spend a lot of time setting up experiments, right? Planning them, doing the cell culture, doing your drug dilutions, running the assay, and maybe not as much time as you should sometimes on the data analysis side of it, because quite often if you can look at something and it looks like it hasn't worked, but if you look at it long enough and you kind of spend as much time on the analysis of the data as you did on setting up that assay, you might actually find that there's something interesting there. So.
No, I absolutely agree. I really loved, in the beginning when you were mentioning you loving running the assays and then sitting down and analyzing the data and plotting it. I love that too. When you're setting up an experiment, you're sitting by the instrument looking at your breath ratio or your fat ratio.
What is the number going to do on the next read?
Exactly.
It goes this way.
Exactly. And then back in the day with your little USB drive, you would take the data from the computer where the instrument was, go to yours. It's 5 p.m. on Friday. It doesn't matter. Just 30 minutes until I put it in my Excel template, then I put it in Prism, and then that feeling of
I can breathe again.
I can breathe. Exactly. I know what happened. I think that's characteristic to everyone or anyone working in the field, or maybe just in science in general.
Yeah, I agree.
All right. Last question I have for you, and I think this is the toughest question. What should we title this episode? That's a very tough one. Yes, I think that's the toughest one. So just to give you a background, and for those who have listened to the podcast many times, I've been doing this for a couple of episodes now. So in the beginning, I used to come up with episode titles based on the conversation I've had. After a while, I just couldn't come up with anything anymore. So the episode title was the name of the guest. So, but the team said, no, Yamina, it has to be very specific. And I said, well, you know what? That's great. But I'm running around. By the time I finish recording, I'm off. And then when the episode has to come out, I have to remember, oh my God, what did we talk about? And how should we make that episode? And that's when the team said, how about you talk to the guests? And then at the end of the conversation, we can together come up with something. And here we are.
Here we are. It's time to get creative.
Yep. It's really about, I think, coming up with a title that would, pique the interest of the audience, but at the same time reflects the direction in which our conversation went, which is why it's hard to do it before we talk.
Yes, that's true. This is really hard. I can understand why you stopped doing it.
I think maybe I can walk you through the overall general sense I've had from our conversation. I think we have a lot in common, and our love for GPCRs and GPCR signaling came very nicely through. I think in your case something that you mention oftentimes is that you're very much fascinated about the intricacies or, the non-canonical type of signaling and how accessory proteins potentially modulate receptor signaling. Yep.
That's a perfect summary. So it's kind of that, and I felt like I was saying organization a lot as well. It's just kind of the intricacies of
Yeah,
I
yeah.
don't know how GPCRs are organized, how they.
Trying to think here. I have my little document open. Organizational intricacies of GPCR signaling. I think this could be really, really interesting. Intricacies of GPCR signaling. I've had episode titles all over the place. We had someone who was now a GPCR scientist, and their grandfather was a scientist, so we called it the gene that skipped a generation. So we can be as.
Oh, that's clever.
I thought it was a really cool title. What else did we have? I'm
Yeah.
trying to look at this here. Up, where is it? Goodness, I have it in front of me. So we had people come up with something. You never know where your GPCR takes you, which I thought was really cool as well.
That is cool.
Know when to walk and when to run. But I think I like our title, Organizational Intricacies of GPCR Signaling.
Yeah, I like it.
Lovely. Awesome. All right. With this, I want to thank you. It was a really fun conversation. I really appreciate your time.
Thanks, Amina. It was lovely to speak to you.
Thank you. Don't go anywhere.
Thank you to Dr. Michelle Halls for such an insightful and grounded conversation. Her work and leadership at Monash are inspiring, along with her commitment to team-based science as a model for how receptor research should happen. Want to help grow the Dr. GPCR community? Here's how. Share this episode with a colleague or a trainee. Subscribe to the podcast so you never miss an episode. And leave a review. It helps others discover the show. Terry's Corner is still your next stop if you're ready to take your pharmacology to the next level. Details on how to access the courses is available in the description of the podcast. At Dr. GPCR, our mission is to advance receptor science through education, community, and by spotlighting tools that move the field forward. And Revity's pH Sense is just that, a real solution to persistent internalization bottlenecks. With pH Sense, scientists can finally track GPCR internalization in real time, even at physiological expression levels, using a simple plate-based format. The reagents are compatible with HTRF readers. Validated on GLP-1R and mu-opioid receptor assays, compatible with HTRF readers, explore it today. Thank you to Celtaris Research for supporting GPCR receptor science through innovative ligand tools. And thank you to the entire Dr. GPCR team who make this show possible. All the links are in the notes of this episode. And because receptors don't work alone, neither should you. Until next time, stay safe.